I'd like to share results of a research study that we've done the last 2 years. Last year when I visited with this group, I showed some preliminary data on the use of chemigation to manage stink bugs in cotton. This year we have finished up that study. We've got 2 years worth of data. We got a lot of replications on things, so I feel pretty firm that what I'm showing you is kind of the final verdict on this particular method. So the idea here is that if we could use chemigation, it provides a lot of benefits to us. We could reduce application costs, we could minimize exposure to applicators, could treat multiple fields simultaneously, and in a year when it's tough to get the sprayer to stand up in the field, this could be a real benefit to us. But in the same vein, as I've mentioned every year I talk about this, the dilution of the insecticide that we're putting out has to be under consideration here. So by airplane we may put out 2 to 4 gallons per acre. By ground sprayer, let's say we put out on average 8 to 10. But by chemigation, you run that little pivot as fast as you can at 1/10 of an inch per acre, you're putting out 2,715 gallons of water. Now I'm still putting out the full amount of insecticide, but it's much more dilute. So the basic objective that I wanted to look at in this study was, are there differences in the efficacy or our ability to manage stink bug populations, whether we put that insecticide out through center pivot irrigation or by ground sprayer? Target pest for this particular study is stink bugs. That's a late season pest. As you all heard me carry on many times before, we get a lot of boll abscission, we get lint staining, reduced lint quality and yield. This is a serious economic pest for us. We see far too much of that when we don't control stink bugs. So the insecticides that we looked at are both labeled for stink bug control in Georgia. Bifenthrin, I'm going to put out the highest label rate that I can, so I'm there at 6.4 ounces. I've got one treatment where I put it out by ground sprayer at 10 gallons per acre. I'm going to do some with an adjuvant and some oil, one without. Then I'm going to do the same thing with chemigation. I'm putting out the same amount of material. I'm still putting out 6.4 ounces of technical, but in this case I'm diluting it in 2715, one treatment with oil, one without. I also picked on Bidrin, which is not labeled for chemigation, which is important to note here. This is more of a research study, but Bidrin has traditionally been our hammer. When we really need to control stink bugs, that's when we pick on dicrotofos. So same treatment structure, by ground sprayer, by chemigation. I've got both lab and field methods we're going to talk about today. In the lab study, we grabbed the insects by the legs, we dipped them in the insecticide, they went into a petri dish, then they went into a growth chamber, and you see I've got these little video cameras there. The video cameras were then pointed at the dishes for about 48 hours, and I could see exactly when that bug flipped upside down. That's what we call knockdown. And then if by chance they went back on their feet and started crawling around, that's recovery. And so I can track all of that for each one of these. And you see there, this is a fair number of bugs we put in there. There's over 1,000 bugs that we dipped and followed these things for 2 days. We've got 3 species that we pulled out of our colony. Today I'm just going to show you the data on southern greens, but southern greens typically show what we saw with the other species. The response variables then will be time to knockdown, the population that was knocked down, the ones that survived, and then the number of visits. And so I've got, in this case, looks like a piece of okra in there. I also sometimes put a snap bean or something, and how many times that bug went over there and fed as a result of that, or while they were in that assay. So this is the time it takes from the time you expose the bugs to the time that they're knocked down, which essentially means they're upside down and they're unable to walk in a coordinated manner. The way my slides are set up, this is the line of demarcation. Everything over here is going to be ground sprayer. So that's the dilutions in 10 gallons of water per acre. Everything on this side is going to be the chemigation dilution. So that's the 2715 number. So what you'll notice is that dicrotofos or Bidrin, very, very quick acting, 10 minutes max. Whoop. Every single bug that we treated with Bidrin, about 10 minutes, and they're flipped over and done. Bifenthrin, it takes longer, but you'll notice that, in a half hour, 40 minutes time, they're all getting knocked out. Now we move to this side of the curve, and you see consistently longer times, an hour, hour and a half, 2 hours time. So we know that we're not nearly as strong in terms of the time that it takes for the bugs to succumb to the poison that we're exposing them to. Well, it's one thing to be knocked down, but the question is, in 24 hours, what proportion of those 40 bugs per treatment were really knocked down? Every single bug that I exposed to that 10 gallons per acre dilution was knocked out. So every single time. There were no cases whatsoever where we dipped them in a insecticide that was intended by ground sprayer and it didn't knock them down. Cannot say the same thing on this side of the puzzle. So at Chemigation, we do a pretty good job on these two, but friends, this is downright scary. Dicrotofos, 30% of them are not being knocked down. So 70% of them, after that treatment, they're still walking around for the full 24 hours. Survival. So this is the total number of bugs that either were never knocked down or they were knocked down, but within 48 hours on my camera, they flip back over and they're walking around and they go take a feeding session on that piece of okra. 0% on the ground sprayer. So what that shows me is that we still have a really good hammer. When we put out our insecticides, be it bifenthrin, be it bidrin, by ground sprayer, I feel very, very confident that we're getting good levels of control. On the chemigation side, this doesn't look too bad with bifenthrin. We're about 90% control there, but this is just flat out scary, y'all. 80% of them are surviving. And I would remind you again that dicrotophos is not labeled for chemigation, but this is a question I get when I give this talk a lot as well. You know, we want to put out whatever the best material we got is, and we know that bifenthrin is not our best material, for example, on brown stink bugs. So that's the reason we've got both of them in there. This is the bifenthrin with a little bit of adjuvant. That's an oil plus an adjuvant or a stabilizing agent, which is often recommended when you're putting stuff out by chemigation. And then this is the number of visits to the food article, so we can look back on that video screen and say, all right, how many times did they actually feed? And the insecticides that are very fast acting, like bidrin, there isn't much feeding that goes on there. If it's slower acting, you get a little bit more, and then these are kind of in the middle there. So in terms of this portion of the study, I would remind you then that as long as we put out what we would say are our ground spray dilutions, where you're mixing the maximum amount of technical insecticide in 10 gallons for an acre of land, we get complete knockdown with zero recovery. That's pretty comforting in my mind. This exposure method, where I was doing this in the lab, where I was actually dipping each individual bug in there, probably overestimates efficacy, because in the field you may not cover the bug that well. You just run over it with a sprayer. You got some fairly fine droplet sizes on there, but I still feel like we're, with a ground sprayer, that's the best that we can do. At the same time, when you put it out by chemigation, I think that you probably do cover the bugs 100%, but the vast majority of the AI that you put in the field ends up on the ground. It doesn't stay up in the canopy where the bugs are. It's going to run down the plant, it's going to end up on the ground, and it's not doing us much good for stink bugs anyway when it's down there on the ground. We did see an increased time to knockdown and significantly more insects recovered when we exposed them to those chemigation dilutions. And the other part of this that really bothers me then is if those insects survive and they go on to reproduce, will their offspring have an even greater propensity to survive when we put out these types of insecticide applications? That's what we call resistance, a change in gene frequency that allows them to survive when exposed to a particular toxicant or a particular type of environment. So that bothers me. I mean, there's a number of red flags here. I want to put that right up there, that this does not appear to be something that we need to be doing on a large-scale basis. Depending on the species that you looked at, when you added that oil, sometimes the efficacy for bidrin increased but decreased for bifenthrin. We really weren't sure why that was. And it changed with stink bug species, so I can't make any blanket statements about how you would handle a field, because every field has a mixed population of stink bugs in it. All right, so then we want to move on to the field component of this. It's one thing to test everything in the lab, but of course we want to be able to apply this in the field, because that's really where the money has to pay. One of the questions I always get when I talk about this is, come on, doc, this is really, really low concentrations of insecticide that you're diluting in, 1,715 gallons of water. How can you be sure that you really got it there? So we set up a system. There's a glass funnel. There's a little collection jar right there. And in every single plot, we measured irrigation water. Went into the funnel, went into the collection reservoir. We took that water back into the lab, and we analyzed that all on a gas chromatograph. So our target, if you put, in the case of bifenthrin, if you mix 6.4 ounces of bifenthrin from the jug with 10 gallons of water, that's going to give you an AI of 2.10 ppm. That's parts per million. So that's my target. In the case of what we collected from the irrigation, on average, I was at 1.82. So I was within 10%. In the case of bifenthrin plus the adjuvants, I actually exceeded it a little bit. That's typical in science. We're not exact. It's going to vary a little bit. There's some error there. But you see that we're generally in the neighborhood of where we need to be. In the case of dicrotophos, we're a little bit over. And in this one, for whatever reason, we were quite a bit over. And I think that it's more difficult to get your calibration right on these chemigation applications because you're putting out so much water. And if there's any surge in the water or anything like that, that affects the dilution rate. So in this case, we had to make an error. I just soon err on the side of putting out more material rather than less. So keep that in mind. I'm at about double the recommended concentration here. Okay, so here are my data. I can't tell you how many hours of work there are summarized in one slide here, so we're going to walk through this fairly slow. This is the 2015 data, starting right at the beginning of bloom, so that's nodes above white flower, going down toward the end of the season. Same thing, 2016, we started about 6 nodes above white flower. Let's pick on the untreated. So the red line is the untreated, and what that's showing us very consistently is that the plots that were in this study, this is a replicated study that we did at Camilla, 8 reps per year, we had more bowl injury. It's off the screen there. I apologize for that. But this is percent bowl injury. We go through there and we take out 10 bowls per plot every week through the course of the season. The percent bowl injury is higher in the untreateds than any other treatment. So that's what we expect to see when we conduct the experiment. Then let's look at the other extreme. On the bottom here, there's a blue dotted line that tends to fall out on the bottom. Here you've got that blue dotted line, and once again, it consistently falls out on the bottom. So that's where we're getting our best protection. Friends, that's dicrotophos or bidrin put out by ground sprayer. Now, does that match what the lab data showed? You're darn tootin'. That's exactly what we saw with the lab data. Same thing showing up in the field. In the middle there, there's a lot more what I would call oatmeal, a little bit more mush. It doesn't separate as cleanly in the field as it did in the lab assay. But I'll remind you that when we do things in the lab, we can isolate all the other variables that are a part of this. We don't have to worry about populations bleeding over from an untreated plot into a treated plot. We don't have to worry about differences based on weather variation. Was the wind blowing when the treatment went out or anything like that. In the field, there's a lot more confounding factors, and we try and take care of that with replication, but it's much more difficult to separate treatments. So what I think sticks out is the top and the bottom. My best and worst treatments are consistent from year to year. The rest of that, it kind of bounces around in the middle. And what I take from that is that chemigation treatment could be beneficial for us if we don't have any other options. If it's a year where we get a hurricane that comes through, dumps 8 to 10 inches on us, and we clearly cannot get a sprayer to stand up, but you need to spray, that chemigation application may be the best that we can do. And we're going to get something from it. I mean, we're showing that we can kill some insects, but it's not as good as putting it out by ground sprayer. These are the lint yield results. This is not quite statistically significant, but the numerical trends are there. My best 2 treatments in terms of yield, that would be bidrin and bifenthrin by ground sprayer. There's the untreated. Numerically, it shakes out at the bottom. And the 2 by chemigation are somewhere in the middle. So it matches pretty well with what we saw in the lab part. Just general results. This is actually my study site. We did this on a lateral. It's a little bit easier to do this on a lateral than a center pivot, but of course the principles are exactly the same. So we would walk this thing across, and I actually went ahead and randomized my plots, and we could turn our sprayers on and off. So we'd walk it across for a treatment, clean everything out, and then walk it back across for the next treatment. So this is basically an all-day affair every time we put out a treatment. Adding those oils, like I said, numerically improved the performance or had no effect, but it really wasn't adding a great deal to us here. I want to make a few comments about the insecticides themselves. We know that bifenthrin, of course, is a pyrethroid. Dicrotophos is an OP. These are contact insecticides. These are not the types of things that go into the plant, get circulated. They certainly are not translocated to the bowl and the seeds, and that's important here. They're not being translocated. The residues are not overly stable. A couple of days, probably maximum that we would expect, and there's a very strong dose response in the lab. So if I mix it stronger, I get more mortality. I think that there may be room for chemigation as a valuable pest management tool, but I don't think it's with contact pesticides. That's what I'm really trying to tell you. So we see reduced efficacy with chemigation. Well, that's kind of like reducing the dilution. The field data, like I showed you, are similar to what we saw in the lab, but they're not quite as clean. And then finally, bifenthrin is labeled for chemigation. You could do that today. Dicrotophos or bidrin is not labeled for chemigation. I want to make sure everybody's aware of that. All right, so I've kind of come to the end of summarizing a long project there. I want to acknowledge the folks in my lab. Everybody pitches in here. This was a graduate student project from a young man named Shing. This is Shing out there in the middle of July with his Superman suit and his hood on and his goggles and rubber boots and rubber gloves. You can imagine how lovely that was in the middle of a cotton field. So Shing did just a phenomenal job. I want to acknowledge funding by the Cotton Commission on this for both years of the project, that this is the kind of thing that we then return. When y'all invest dollars into the research program at UGA, this is the kind of results that we return from there. So I'm going to stop speaking. Jason's going to come up and load his talk, and I'll be happy to address questions in the meantime.